Jens, do you have anything to share with us?
There are lots of solutions or lots of approaches to solubility improvement, and that's probably a challenge for everybody to understand the value of specific methods and what they can bring to your specific challenge. And well, I think it was more or less the finding that there are so many methods.
Different methods, different problems. Okay, anything from this side, from the front here?
Well, I think that I'm working in a real early stages, and I think you can start with driving your chemistry to be as soluble, deliver as soluble compounds as possible. So
yeah,
I think it starts
not
there.
ending up with lead candidates with severe solubility
Yes,
problems.
yes. Because it's, I mean, I think certainly in earlier stages it's not like, okay, we have now the most active drug or compound, let's say, we throw it over the wall and the drug, the guys that have to come up with the molecule or the formulations itself, they will solve all the problems.
Yeah.
Which is not the case, I guess.
No.
So that's my...
It's a good point too. Anybody from this side?
I just would like to agree with that. I have had about over 25 years of experience dealing with medicinal chemists developing brick dust,
yeah,
and throwing it across the wall. Yeah. But however, I think we have to accept the reality that when they are screening compounds, they look for the most effective and least toxic. And you cannot always have everything, including good solubility. So it is down to formulators to try and resolve that issue. But again, solubilizing insoluble drugs is not impossible, but when you arrive at a solution, it depends on the route of administration. For example, twin ATs and carbopols are very good solubilizing agents. But if you were to use them for intravenous application, you often end up with anaphylactic reactions. That's because they release histamine, and if you are giving these stuff at high concentrations intravenously, they end up in the liver, and liver is a very big source of histamine.
Yeah.
Therefore, you get anaphylactic reactions, and it actually destroys your product. So you have to be careful about the route of administration as well as the application. And in oral administration, you have got a wider choice of solubilizing agents. It's not always impossible, but this is a problem that formulators have to live with day in and day out.
Yeah. Okay, anybody else from this side? No, nobody else. Okay, good. I'm going to touch upon some of these in my presentation. There will be more slides. The rest, you can be calm. Administration routes. So you can use mesoporosilicas for all applications, parenteral, there are some vaccine applications. We have some data on dermal application and also nasal. However, I'm going to talk about oral here today. There are different problems to solve. Of course, the enhanced dissolution is one. The protection of shelf life stability, being trapped inside a pore, you can have mitigate, for example, hydrolysis or oxidation or sensitivity to light. Of course, there is a whole set of projects that we do with bioequivalence, where we just mimic the curve, being able to tailor our pores, the particles, the system. We're pretty good at mimicking existing drugs. And of course, sustained release. That is usually not only with silica, but also with the coating. but I'm going to talk about improved solubility. We get a lot of questions of how we get the drug, how we load. So basically we dissolve the drug into an appropriate solvent. So it's dependent on the API, not upon the silica. The silica is very resistant. And then, since the pores are very narrow, the capillary force is very strong, the solvent and the API is sucked into the pores, and then we evaporate the solvent, and we have data showing that there is no solvent remaining, there's no solvents left in the silica matrix. So being very regular and very porous, we can load from something from 35 to 48 weight percent. And then so it's trapped inside. And it's actually, I'm going to take you through the three factors basically that we call lock-in, spread out, and jet beam, which is why we increase the solubility. So basically we lock in, so we're talking about dust bricks here, crystalline APIs that crystallize, or molecules that crystallize or recrystallize. So basically they are loaded in an amorphous state into the silica matrix. The pore being very narrow, actually it's the lack of physical space that keeps the drug amorphous, and it stays amorphous for years. The spread out effect is of course that since it is in, well, I mean, silica is very hard and it's very easy to tablet. So once it gets into the gut, let's take a tablet. When it gets into the gut, it's basically the silica particles are distributed very evenly in the liquid, which makes basically a very homogeneous concentration. You don't get this super high concentration near the tablet that could be the case with our other technologies. And the last thing is basically we've studied this in live NMR, where we actually this very narrow pore has the driving force of the capillary driving force is very strong. So the particles are basically beamed out in the liquid. And we talked about, I was there on one of the presentations of nanomilling, so they get down to 150 nanometers. Us having a pore of 3.4 nanometers has chunks of API of a single nanometers size coming out. So it's so the contact area, of course, is huge, having such small particles coming out of the pores. So that's the three principles basically: lock-in, spread out, and jet beam. This is Thembi Ngubane. She was a local celebrity in South Africa. She was infected with HIV at the age of 16. She had her first child with another HIV positive man when she was 21. She was on a drug called atazanavir during her pregnancy, and the child is HIV negative. She died of TB when she was 24. So this is the story about atazanavir. Antiretroviral blockbuster by BMS, selling for $1.7 billion per year. Quite tricky molecule, and its solubility is very pH dependent. So low solubility, nice dissolution, neutral pH, horrible. So of course you mustn't co-administer with proton pump inhibitors, but there's actually one other. So being a patient with a chronic disease, you need HIV patients, especially non-naive, they take proton pump inhibitors much more frequently than the rest of the population. There is also another unmet medical need, which is in the sub-Saharan Africa. Malnutrition also have an effect on the pH in the stomach, elevating the pH. So there is actually a problem also there where this drug is widely distributed that has lack of efficacy. So what we did was in a rat study, we co-administered proton pump inhibitors with atazanavir. So as you can see, this is the graph of the free drug, and this is loaded in our particles. So you can see the difference in both Cmax and AUC. It's in this case fourfold. We have similar data on other drugs as well. And this is, so we show with XRD that this is the image that shows that in the pore it actually is amorphous. So, coming to the end of my presentation, so we have life cycle extension projects like at the Zanavir. We're into generics, which is actually about, mostly is about bioequivalence projects, tailoring the particle system so that it matches a reference product. Of course, it's a pipeline expansion. We have one product with a company where they've tried, it's a very nice data on preclinical, however, they can't get into the maximum tolerated dose to move into the clinic because the molecule doesn't dissolve. So we've been able to help them with that. I guess that our conclusion is that this is for the, not for all polysolubles, and not even for all crystalline drugs, but for the ones that are really tricky. And of course, we have our own assets that we put ourselves. We currently have one in obesity, and we have several others lining up. But I think the conclusion, I've been the CEO of the company for 1.5 years, is that we need focus. So we're focusing on oral delivery and enhanced dissolution. And that was the end of my presentation.